right, so let's finish this nervous system lecture. And we're going to start with the glial cells, which we also call neuroglia. And we find glial cells in both the central nervous system and the peripheral nervous system, but we're going to have different ones in both places. So pay attention to where each kind of cell occurs. Glial cells are much smaller than neurons. They're those tiny little things that we saw on that slide of the neuron smear. They are capable of mitosis, and so we'll talk about how that affects their function. They're going to help the neurons do their job. So in that way, they're kind of like the satellite cells that we see in skeletal muscle tissue. They're a little different because each kind of glial cell has a different function of its own, but they're also going to help the neurons. And then we can tell them apart by their size, what they have inside their cells, the presence of different processes on the cells, all kinds of things. So this is an overview showing the different kinds of glial cells. So to orient you, this is a neuron here. This is a multipolar neuron. Here's another one here. And this is inside the brain. So here's our cross section of the brain. You can see they took one little part here. So we have the neurons. You can see we've got myelin here. We've got a myelin sheath. But these cells look very different from the Schwann cells that we see in the peripheral nervous system. They're called oligodendrocytes. Remember, dendro means tree. Cyte, of course, means cell. And oligo means several. So these are cells that have several branches on them. Not tons of branches, but several branches. They're going to create the myelin sheath in the central nervous system. We also have little tiny microglial cells. We have these others called astrocytes that are kind of star-shaped. They're going to support the capillaries. Then we have these things here called ependymal cells, and they're going to make this fluid that surrounds everything in the central nervous system. So let's start with the astrocytes. These are the most abundant glial cells in the central nervous system, and they do make up about 90% of the nervous tissue in some parts of the brain. So this again is where we get that you only use 10% of your brain idea. In fact, you use your whole brain, but only about 10% of it is actually neurons, and the rest is glial cells, and in some places, actually more than 90% of it is astrocytes. So astrocytes, they're called astrocytes. Astro means star. So the reason they're called astrocytes is because they have all these projections on them. The projections themselves aren't branched. And each one has this little expanded area at the end, which is called a little perivascular foot. And they wrap these extensions and these little feet around the capillaries, and then some of the others, they grab onto nearby neurons. So they're going to help hold the neurons in place, and then with the perivascular feet, they're going to create a barrier around the capillaries that go through the central nervous system. Altogether, these perivascular feet make what we call the blood-brain barrier. And the blood-brain barrier is the thing that regulates what the fluid is that's around the brain. So think of this as the blood-brain barrier. Think of it as an extra layer of protection around the brain to protect the brain from materials in the blood. Might be okay running around other places, but you really don't want them to have an effect on the neurons. So the blood-brain barrier is going to control what substances can get into the brain and the spinal cord and what can't. The astrocytes also help regulate synaptic transmission. So in their communication with neurons, they're going to help regulate synapses. We are not going to talk about that in this class, but that's something you can learn about elsewhere. They're going to strengthen and organize the tissue because they hang on to lots of different things. And then when neurons are damaged, astrocytes can help to replace those. So they do have a really big important job. And then during fetal development, they're going to help build the brain in the fetal development. Now, ependymal cells are ciliated cuboidal epithelial cells. And they're only found in the central nervous system, and they're only found in certain areas in the central nervous system, and that is around the central canal of the spinal cord, which is actually a little hole that runs through the center of the spinal cord and is filled with fluid. And then there are these spaces inside the brain called vesicles. And these ependymal cells line these spaces, or are in certain sections of these spaces. And a bunch of them together is called the choroid plexus. And what they do is they are going to filter the fluid, the extracellular fluid, and create this fluid called cerebrospinal fluid. The cerebrospinal fluid is going to bathe all of the surfaces around and within the brain and the spinal cord. And it's also going to support the brain. So the brain actually floats within the skull on this tiny, tiny layer of cerebrospinal fluid, or CSF. And because these cells are ciliated, they're going to create waves that move the cerebrospinal fluid around. And that's also going to help carry waste away from the brain. So we're going to come back to that later, but that's what the ependymal cells do. Now, microglial cells, they're really tiny. They have a few little branches on them. And these are going to move around through the central nervous system. They're going to be immune cells. When there is an infection in the central nervous system, they are going to do mitosis so that they can phagocytize dead cells or damaged tissues and clean up any issues, any problems, any dead cells or any damaged tissue within the central nervous system. So they are phagocytes, and they do move through your central nervous system looking for damage, looking for abnormal cells, and then engulfing them and getting rid of them. All right, now oligodendrocytes. Oligodendrocytes make the myelin sheath in the central nervous system. So in the peripheral nervous system, the myelin sheath is made by the Schwann cells, or the neurolemocytes. The oligodendrocytes are a little different because they have processes that they stick out to several axons and wrap around to create the myelin. So the difference is in the peripheral nervous system, each Schwann cell wraps itself around the axon, its whole self. In the central nervous system, each oligodendrocyte helps wrap a number of axons, okay? And remember that all of that together is called myelin. And myelin is just layers and layers of plasma membrane from these glial cells that insulate the axon. And because they provide insulation, they actually make the signals travel faster down the axon. So they speed up signaling in this nervous system. Now, those were all central nervous system glial cells. Now, in the peripheral nervous system, remember I told you we have different glial cells in the peripheral nervous system. So we do have satellite cells, just like around the skeletal muscle tissue. We have satellite cells around cell body, in ganglia. So this is the posterior root ganglion, which is right next to the spinal cord. So we have axons from sensory cells. The signals come into these cell bodies to be integrated, and the satellite cells insulate the cells from each other, but also provide some support and communication. And then the signals go from the cell body back out. So these are satellite cells supporting these cell bodies, just like the satellite cells in muscle tissue does. And then we have the Schwann cells, or the neurolemocytes. And you can see that here we have one cell that wraps itself around to create the myelin sheath, and then another cell, and another, and another, and another, and another. Each cell wraps its whole self around the axon. So this is an MRI that shows a glioma, and you remember that oma means tumor. And so a glioma is a tumor of the glial cells. And this is a more common form of cancer than a myeloma or a neuroma. And the reason that is because glial cells do mitosis and neurons don't. So it's much easier for the mitosis system in use to misfunction than in, for example, a neuron that's not using the mitosis system at all, for that to then start turning on and then go abnormally copying cells. So the neurolemocyte starts to, this is how the myelin sheath is formed. So this is a neurolemocyte, but the oligodendrocyte is going to do the same thing, just not the whole cell is wrapping itself around. So here's the axon, here's the neurolemocyte. During fetal development, it starts wrapping itself around, and then it just keeps wrapping. The cytoplasm form layers, and then eventually the cytoplasm all gets smushed to the side, and it's just plasma membrane, wrapped layer upon layer, until we end up with a cell that cytoplasm is all smushed to the sides, nucleus is smushed to the side, and we have layers and layers and layers of plasma membrane creating what we call the myelin sheath. And you remember, of course, that the plasma membrane is made up of phospholipids. So this is basically just a layer of fat, which provides excellent insulation. It also means that when we look at areas of the body that have lots of myelin, what we see is the tissue looks white. So if we come back up here and we see this cross-section through the brain up here on the left, we have this outer layer, which we call the gray matter, and then the inner layer we call the white matter. The gray matter is mostly cell bodies that aren't myelinated. You see, the myelin doesn't go to the cell body. The white parts, the white matter of the brain and the spinal cord, is myelinated axons, and it looks white because the myelin is a layer of fat. So what we're seeing when we see that white matter is we're seeing all of this fat in the myelin sheaths. So here's the distinction between the oligodendrocytes in the central nervous system and the neurolemocytes or Schwann cells in the peripheral nervous system. So they're both going to make myelin on axons, but the oligodendrocytes, one cell produces myelin for multiple axons. In the peripheral nervous system, the Schwann cells or neurolemocytes, each cell wraps its whole self around one axon. So each cell only protects one axon. In both cases, in both places, the myelin sheath is made up of plasma membrane from many, many cells. And then the gaps between the myelin pieces are called the neurofibril nodes or the nodes of Ranvier. And that's the same in both places. So that's how you distinguish those two. Now, if you have a condition where your neurons, your glial cells don't make myelin correctly, or where your body starts attacking the myelin cells, then you would expect that signals are not going to travel down these axons at the right speed, because remember, the myelin insulates the axon, and the signal goes faster down the axon because of the myelin. And one condition that is caused by a lack of myelin is multiple sclerosis. And this is Selma Blair, an actress who has multiple sclerosis. And this is her last year or 2 years ago at the Vanity Fair party for the Oscars. And it was her first time in public after receiving the multiple sclerosis diagnosis. And one of the painful things about multiple sclerosis is that the symptoms come and go. They're not usually consistent. They're very imprecise is not the best word, but it's hard to tell what's going on. And the way you end up diagnosing multiple sclerosis is that you eliminate all the other possibilities. So people start having, they become klutzy, they get headaches, they get numbness, and some days they're much better than others. So the symptoms do come and go, which is one of the things that's very heartbreaking about multiple sclerosis. So this is Selma Blair, and I want to show you a short clip of her talking to the paparazzi. I do not want to show you that. So she looks amazing. And a lot of disability rights people were really, really excited that she used her cane because, you know, there's a lot of hard feelings about canes. And so you can see she looks amazing. And so now you see she's going to walk, and you see what a hard time she's having. And you can see that she's having such a hard time walking, and it really, even the paparazzi are affected by it. So it is a really heartbreaking condition. Oops, that's not my one. There we go. So that's multiple sclerosis, and it's because of a lack of myelin. Because of the lack of myelin, the signals going back and forth in the body aren't going at the right speed, and so the body is not able to communicate correctly the sensory and integrate the sensory and motor information correctly. And so it does make it very hard to do simple things just like walking. So there are also things that we call unmyelinated axons. They aren't completely unprotected. They do have a little bit of extra plasma membrane around them, but they don't have the really thick layers of myelin like the myelinated axons. So in this case, here's our neurolemocyte, and instead of wrapping itself many, many times around one axon, it puts out a little bit of plasma membrane around a bunch of axons. Just kind of gives it a little wrap like that. And when we look at these on an electron micrograph, so this is 60,000 times normal size, this is our axon, and you can see the layers of myelin in here. And if I zoom in, you can see it really clearly. So you can see all these layers here, that's layers of plasma membrane. And then out here, we can see the cytoplasm of that myelin-making cell. And then if we, come back down. Where do we go? Oops, sorry, went way too far. And then up here, we can see unmyelinated axons, and you can see this extra layer of plasma membrane. So we've got the plasma membrane of the cell there, and then out here we've got this extra layer kind of going around and adding some protection for all of these axons that are what we call unmyelinated. So these are not as well insulated, and signals are going to travel through them more slowly. So with myelin, because each of these myelin cells, each piece of myelin, insulates the signal, When we measure the signal going down an axon, we don't see it. We can't measure it while it's passing through these spaces. We can only see it when it gets to these neurofibril nodes, these little gaps. So when we're measuring, it looks like the signal jumps from here to here to here to here to here to here to here. So it looks like it jumps from one node to another. That kind of movement is called saltatory conduction. And it comes from this word saltatory that means to jump. And so here's an example to help you remember that word saltitation. Here is an animal doing saltitation down the beach. Also, so freaking cute. Right? See, jump, jump, jump. Saltitation. There you go. Now, when all of that has, so far we've just been talking about glial cells and neurons individually. We mentioned nerves just a little bit, and now we're going to come back to the nerves and talk about the structure of nerves. And as you can see, this looks really similar to the structure of a muscle. We've got a bunch of small things wrapped in an endo something, and in this case, it's the endoneurium. Remember, we had the endomysium in muscle. Now it's the endoneurium. It's going around a neuron. So the endoneurium goes around the myelin sheath if the cells are myelinated. We have a bunch of axons in a fascicle, and the fascicle is surrounded by a perineurium. Several fascicles together make up a whole nerve, and that nerve is surrounded by an epineurium. And the perineurium is going to also fill in the spaces between the fascicles and provide space for those blood vessels to run through there, to provide blood for the neurons themselves. When we look at these on a regular microscope slide, we can see the neuron here with the myelin sheath around it. The axon is this dark line in the middle. And then this thinner stuff around it, that's the myelin. Okay, and then the nucleus is the nucleus of the myelin cell. When we look with an electron micrograph, we can see the fascicle is made up of a bunch of axons. This layer around that is so the endoneurium is here, this is the perineurium, and then we have blood vessels running through that perineurium. All right, so what happens if you damage a neuron? A lot of neurons can actually regrow a portion of the axon if it is damaged. Now, this isn't always possible, and in some parts of the body it's not. We'll come back to that when we talk about the spinal cord. But basically what happens is if there's damage to the axon, then the proximal end, closer to the cell body, seals off and swells up. And then past that, the distal end of the axon and the myelin sheath, they both disintegrate. The neural lemma, which is the plasma membrane of the neuron, and the endoneurium together form a tube called a regeneration tube that reestablishes connection with, in this case, the muscle. And then within that tube, the axon regenerates, and then the myelin regenerates as well. And then finally, the innervation, or the nerve signal to the effector, is restored. Okay, so that's how you can regrow nerves. If you've ever had like a really deep cut and you suffered some numbness from that, that damaged those neurons, and then if that numbness went away eventually, that was through this neuron regeneration that was happening. In the central nervous system, there's a couple things that happen. One is that the oligodendrocytes don't release growth factors. So that's a chemical that helps with the healing. It helps with all this regrowing. The other thing is there's just too many axons in one place, and the action of the immune response in cleaning up the damage often leaves scars. And so because of that, especially in the spinal cord, if we have damage to too many of the axons in the spinal cord, then often it is permanent because there's a scar and the neurons literally can't send signals past that scar. So that's why damage to your spinal cord can cause paralysis. So when we have spinal cord injuries, the axons above the injury are still fine, work fine. Everything below the injury, the axons will often die and the muscles then become paralyzed. So depending on the site of the injury, depending on which vertebra is involved, that makes a difference of how much of a person's body is paralyzed. I am going to skip past these two for today. Go ahead and watch that. This is a guy who's actually broken his neck a couple of times. He's a little nutty. I love him to death, but we will come back and talk about him when we do the spinal cord. All right, so let's talk about synapses. A synapse is a connection between two neurons. So we have one neuron, in this case, a multipolar neuron. The signal comes in the dendrites here, through the cell body, down the axon, out the axon terminals to the synaptic knobs. And then those synaptic knobs connect with either the dendrites or sometimes the cell body of another cell. And we use the clever terminology presynaptic neuron for the cell that comes before the synapse, and postsynaptic neuron for the cell that comes after the synapse. And you remember the synapse is actually the connection between the two cells. So we had a synapse between the motor neuron and the skeletal muscle cell when we did the muscles. So we have the signal comes from the cell body down the axon, and once it gets to the synapse, then there is in fact a cleft, a space which we call the synaptic cleft, before the postsynaptic cell. And we'll talk about how this signal happens. So synapses are these connections between neurons. The nerve impulse travels from one cell to the other, from the presynaptic neuron to the postsynaptic neuron. And the space between them is the synaptic cleft. And except in the heart. The gap junctions in the heart are actually considered electrical synapses. We are not going to use synapse to mean electrical synapses, but I'm just throwing that out there so that you understand that that is a term that people do use. We are going to refer to chemical synapses, which are the ones that we have in the nervous system. So the action potential, remember there's that change in the charge inside the cell as a result of opening and closing sodium and potassium channels. That action potential travels down the axon terminal until it gets to the synaptic knob. That change in charge, that voltage change, opens channels on the synaptic knob that are calcium channels. Okay, so these are voltage regulated or voltage gated calcium channels. When those open, calcium ions flood into the neuron. So the neurons do maintain a concentration gradient, high calcium levels outside the cell, low calcium levels inside the cell, so that when these channels open, calcium rushes into the cell. The calcium causes exocytosis by vesicles in the synaptic knob. Now it turns out that this is actually a really, really complicated signaling process, so we are not going to talk about it. Just know that calcium goes in, exocytosis happens. And you remember that these vesicles have some kind of neurotransmitter in it. In this case, it's acetylcholine. You remember that's the one that we used with muscle cells, and it's used in other places as well. So voltage-gated channels open, calcium rushes into the neuron at the synaptic knob. The synaptic knob does exocytosis of vesicles and releases a neurotransmitter. Now there are a bunch of different neurotransmitters, so it's not always acetylcholine. The neurotransmitter is released. It binds to a receptor on the postsynaptic neuron, and that opens a channel that allows something to go into the postsynaptic neuron. These are usually sodium channels. Not always, of course, it's always more complicated. But these are usually chemically gated sodium channels. So they're chemically gated, meaning they open because a chemical has bound to the channel. Once that neurotransmitter binds, then the sodium enters the cell. The sodium floods in, and that changes the charge inside the postsynaptic cell. Now we can have an action potential in the postsynaptic cell. The flood of sodium ions then causes that, and let's look at this. Let's see. Oh, okay, so this is Mouse Party. Am I going to have space for this? Let's see if we have space for Mouse Party. Oh, Mouse Party is not playing. Oh, no! It's from the... Okay, shoot. All right, I will find... see if they have a replacement for Mouse Party. Mouse Party is a really good way to see like how these chemical gated channels work, and it shows lots of different neurotransmitters that are at work in the brain and how different drugs affect those. So it's a really fun interaction. I'll show you. I'll try to see if they have a replacement for it. So several different kinds of diseases can be caused by degeneration of nervous system cells. We talked about multiple sclerosis, amyotrophic lateral sclerosis, ALS, is the disease that Stephen Hawking had, and it is also a neurodegenerative disease. So because of the way that neurons work and because of the neurotransmitters released by different cells, toxins often work by either blocking receptors or acting like a neurotransmitter. So curare and atropine are chemicals that come from plants. They block the receptors for acetylcholine. So if acetylcholine is released and something else is already attached to that receptor, then the acetylcholine can't bind to the receptor and open the channel. And so then the person is paralyzed because literally the acetylcholine can't be released to start the muscle contraction. Alpha bungarotoxin comes from this snake. It also blocks receptors for acetylcholine. It is used in research to tag neuromuscular junctions so that you can determine, like, for example, how many neuromuscular junctions a person has. Tetanus blocks the neurotransmitter release. So tetanus is a toxin that comes from a bacterium that lives in the soil. And if you get that bacterium in your body, then the bacterium starts to release tetanus. It blocks neurotransmitter release, and you will die of paralysis. Botulinum comes from another bacterium, and it also blocks neurotransmitter release, and it paralyzes muscles. And in a very, very dilute form, that is Botox. That's what is injected in Botox. Botox literally paralyzes the muscles, and those muscles, when they're contracted, cause wrinkles. So then you paralyze them, they relax, and they make wrinkles go away. You also can't make facial expressions because your facial muscles have been paralyzed. Nerve gases block the recycle of neurotransmitters. So let me say one more thing about this. So let's go back here. So these neurotransmitters, for example, acetylcholine, they're going to be released into the synaptic cleft. They're going to bind to the receptor. They do not enter the postsynaptic cell. Something else, sodium ions, enter the postsynaptic cell. Instead, the neurotransmitter gets pumped back into the presynaptic cell. Okay, so there are little pumps along here that are going to take the neurotransmitter, the acetylcholine, and pump it back into the synaptic knob. So if you block that action, then the neuron can fire once, but then it can't fire again because it runs out of neurotransmitter. So that's another way that toxins work. So before I finish, I want to talk about neuronal plasticity. And this is the ability of the brain to grow and reorganize its synaptic connections. And this is really, really important in fetal development and then in the first few years of life. So we can actually see synapses growing in the fetal brain and then in the infant brain up to about 2 years. And at 2 years, the brain actually has more neurons and more synapses than it needs. And so for the next few years after that, the brain actually prunes away synapses, leaving the ones that are most valuable. And so this is why they say that these early years of development are most important for children, because what gets pruned away leaves basically a blueprint for what the brain is going to be able to do later. So if you prune away too much in these early years, then it makes it that much more difficult to build new connections later. So we do know that brains can change and that certain brains, that when you use a part of the brain, that you get more connections in that part of the brain. So there was a study done of London taxi drivers versus London bus drivers. If you think about a bus driver, they drive the same route with the same stops every day. So you got to learn the route, you got to know where to stop, but you don't have to know a huge volume of stuff. London taxi drivers, on the other hand, have to know the entire city, every single block in every neighborhood. And in order to become a London taxi driver, you have to sit for something they call the test. And to do the test, you sit in a chair and someone says, how do you get from here to here at this time of day? And you have to speak the entire route from beginning to end and estimate how long it would take. So you have to have this incredible spatial knowledge. And so what we see when we compare the brains of taxi drivers versus bus drivers is that the bus drivers have very small areas for spatial memory, whereas the taxi drivers have much larger spaces because they're literally storing more spatial information. We also, this is a study that was done in rats, where rats were learning a new task. And they did brain scans at the beginning and then after 1 day and after 4 days. And what they saw was after 4 days where the rats were learning new skills, they actually saw new dendritic spines appearing. This means that the rats, as they learn a new task, are actually putting on new dendritic spines in their brain. So there's one final thing. This is an experiment to look at the effect that an enriching environment has during the early part of a rat's life. So we have this one rat who for the first 30 days was raised in a really boring environment. And then for the next 30 days, some of the rats stayed in the boring environment and some others went to this really well enriched environment where they had toys to play on and a wheel to run on and all kinds of stuff. Viva Rat Vegas. What we see is the unenriched rats, their brains are actually simpler than the rats raised in an enriched environment. So those rats who had things to do, things to play with, wheels to run on, they actually made more connections in the brain than rats who didn't have those things. So that's why, with children, you need to make sure they get lots of different kinds of enrichment. They need to use their body and not just look at screens. They need to build up the synapses to control the muscle movements so they can develop fine motor movements by doing things like playing with blocks and coloring. And yes, coloring inside the lines is actually a skill that children need to have because it shows small motor skills. And all of that is really important because of the effect that has on the growing brain. But what we also know is that mature brains can grow more connections. So it's not a permanent deficit, usually, as far as we can tell. We know that adults, even very, very old, can grow more connections, and that's what you're doing right now. So take care of your brain, eat good food, get lots of sleep, build new neurons, build new synapses, build new dendritic spines. All right, so that's the end of that lecture. Thank you for sticking with me today. I appreciate it, and I will see you next time. Take care.